Welcome everyone to another lecture in general biology. We are now looking at chapter 5 of Campbell Biology, the structure and function of large biological molecules. All living things are made up of 4 classes of large biological molecules: carbohydrates, lipids, proteins, and nucleic acids. Macromolecules are large molecules and are complex. Large biological molecules have unique properties that arise from the orderly arrangement of their atoms. What we're looking here is an example of a protein macromolecule with alpha helixes, those purple spirals and beta sheets, those flat plates in blue and green. Macromolecules, they are polymers built from monomer units. A polymer is a long molecule consisting of many similar building blocks. The repeating units that serve as building blocks are called monomers. Carbohydrates, proteins, and nucleic acids are all polymers made from monomeric units. Enzymes are specialized macromolecules that speed up chemical reactions, such as those that make or break down polymers. A dehydration reaction occurs when 2 monomers chemically bound together through the loss of a water molecule. A way of looking at this is one monomer loses an OH, the other monomer loses an H, so that a chemical bond forms between those 2 monomers, and then the OH and H come together to form a water molecule that leaves. Polymers are disassembled back into monomers by hydrolysis, a reaction that is essentially the reverse of dehydration, where a water molecule is essentially sent into that chemical bond, and then one monomer leaves with an OH, and then the other monomer leaves with a hydrogen. As seen here in a dehydration reaction, essentially a hydrogen hydroxide group, they leave forming water, and then what is left is a chemical bond between those two monomers, or the polymer and then the unlinked monomer. Hydrolysis is simply returning those components of water to split up 2 units. A cell has thousands of different macromolecules. Macromolecules vary among cells of an organism, vary more within a species, and vary even more between species. A huge variety of polymers can be built from a small set of monomers. Looking at carbohydrates, those are sugars and the polymers of sugars. The simplest carbohydrates are monosaccharides, or simple sugars. Those are the monomers of carbohydrates. Carbohydrate macromolecules can be built from 2 monosaccharides to form disaccharides, or multiple monosaccharides to form polysaccharides. Monosaccharides have the molecular formula that are always or usually a multiple of 1 carbon, 2 hydrogen, and 1 oxygen. Glucose, for example, is C6H12O6, and the most prolific and most common monosaccharide. These can be classified by the location of the carbonyl group as either aldose or ketose, coming from aldehyde or ketone. The number of carbons, and as well as by the number of carbons in the carbon skeleton, 6, 5, or more. Here we have just different types of carbohydrates. What we're looking at in the middle and bottom, pentoses, which are 5 carbon sugars, hexoses, which are 6 carbon sugars. Many common ones being glucose, galactose, and fructose, where glucose and galactose are both aldoses, where the terminal carbon has a double bonded oxygen, whereas fructose is a ketose, where one of the middle carbons has the carbonyl group. Trioses are another example of just 3 carbon sugars. Interesting to note is that glucose and galactose are simply enantiomers. Same chemical structure or formula, but one carbon in the chain has hydroxide on one side, whereas between glucose and galactose, where it's on the other side. Though often drawn as linear skeletons, in aqueous solution, many sugars actually form rings. Monosaccharides serve as major fuel for cells and as raw material for building molecules. So in solution, or in their normal structure, usually glucose or galactose, they exist as a 6-member ring, where 5 of the members are carbon and 1 is an oxygen. Now, a disaccharide is simply formed when a dehydration reaction joins 2 monosaccharides specifically. This particular covalent bond between monomers of a saccharide is called a glycosidic linkage or a glycosidic bond. Dehydration to form the disaccharide maltose is just 2 glucose sugars coming together. And then dehydration to form sucrose is the glycosidic bond formed between a glucose monomer and a fructose monomer, where the fructose is the 5-member ring, where one of those members is oxygen and 4 are carbon. What we see here, the 1,4 glycosidic linkage, that's between carbon 1 of glucose and carbon 4 of glucose. Whereas the 1,2 glycosidic bond is between carbon 1 of a glucose monomer and carbon 2 of a fructose monomer. Polysaccharides, the polymers of sugars, have storage and structural roles. The architecture and function of a polysaccharide are determined by its sugar monomers and the positions of its glycosidic linkages. Some that are used simply for storage include starch. It's a storage polysaccharide found in plants and consists of numerous glucose monomers. Plants store excess starch as granules, which chloroplasts within chloroplast organelles and other plastids. The simplest form of starch is called amylose. Essentially, what we're looking at is an example of storing multiple glucose monomers as a polysaccharide that will eventually be used later. Glycogen is an example which has many different branches to store many types of glucose. Now, as found in potatoes, there is amylose, which are unbranched glucose chains, and then there's amylopectin, which has a little bit branching. But in animals, where we can store things as glycogen, there is numerous branching. Glucose is used for producing ATP later in biochemical reactions. So storing excess glucose is useful for when it's needed on reserve, when there isn't any available immediately. Glycogen is a storage polysaccharide in animals, sort of like starch for plants. Glycogen is stored mainly in the liver and muscle cells. Hydrolysis of glycogen in these cells releases glucose when the demand for sugar increases, but it's not freely available yet in the diet. Now, as far as Going beyond storage, using polysaccharides for structure, there are polysaccharides called cellulose, and that is a major component of the tough cell wall in plant cells, aiding to their rigidity and structure. Like starch, cellulose is a polymer of glucose, but the glycosidic linkages differ. The difference is based on the two rings forming glucose, either alpha or beta. Getting into those alpha and beta linkages, what we see here is isomerization between alpha glucose and beta glucose, where the difference is how is that primary alcohol facing? Is that primary alcohol facing down, or is it facing upwards? That will owe to either alpha glucose or beta glucose. Now, as far as the ring structures, joining of two glucose molecules you have a 1,4 linkage. It's however in B, the 1,4 linkage of alpha glucose monomers. The glycosidic bond appears to be up and down, whereas in revolving alpha beta glucose structures, those glycosidic bonds are all facing the same way. Or no. A is alpha and beta glucose, the top structure. B, as in starch. The linkage between alpha glucose monomers forms a 1,4 glycosidic bond, where they're all facing down. And then the cellulose 1,4 linkage of beta glucose monomers has an alternating glycosidic bond, which essentially looks like those monomers are flipping as you go down the chain. Starch, which is the alpha configuration, is largely helical, whereas cellulose molecules have beta configuration, which are straight unbranched chains. Therefore, that's where we get the terminology alpha helixes and beta sheets. Some hydroxyl groups on the monomers of cellulose can even hydrogen bond with hydroxyl groups of parallel cellulose molecules. Enzymes that digest the starch by hydrolyzing alpha linkages can't hydrolyze beta linkages in cellulose. The cellulose in human food passes through the digestive tract as insoluble fiber. Some microbes use enzymes to digest cellulose that we otherwise could not. Many herbivores, from cows to termites, have symbiotic relationships with these microbes to help digest this material. Chitin, another structural polysaccharide, is found in the exoskeleton of arthropods. Chitin also provides structural support for the cell walls of many fungi. Structural form of chitin monomer includes our 6-member monosaccharide, as well as a amide group that's carbonyl bonded to an amine. Lipids are the one class of large biological molecules that does not include true or typical polymers. The unifying feature of lipids is that they mix poorly, if at all, with water. They're very hydrophobic. Lipids consist mostly of hydrocarbon regions, which are hydrophobic. The most biologically important lipids are known as fats, phospholipids, and steroids. Fats are constructed from two types of smaller molecules, a glycerol and then 3 fatty acid side chains. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon, and then the fatty acid consists of a carboxyl group attached to a very long chain of carbon and hydrogen, such as shown here. Now, similar through dehydration, a fatty acid can link up with a glycerol with either of the carbons, oxygens in a glycerol, and that's how we get triglycerides, such as the example here, a triglycerol. The bond is called an ester linkage between a carbon that is double bonded to an oxygen and single bonded to another oxygen. And that is an example of an ester. Fats separate from water because water molecules hydrogen bond to each other and exclude fats. If in a fat, three fatty acids are joined to glycerol by an ester linkage, creating a triglycerol or a triglyceride, the fatty acid in a fat can be all the same or 2 or 3 different fatty acid chains. Fatty acids vary in length, and that is the length of the carbon backbone, and in the number of and locations of double bonds. Saturated fatty acids have the maximum number of hydrogen atoms possible, and no double bonds. Unsaturated fatty acids essentially have a fatty acid side chain that has at least 1 double bond, maybe more. When there are no double bonds, these fatty acid chains can be very straight and collapse in on each other, causing them to be very stuck and very hard for the body's enzymes to reach. Whereas when there is an unsaturated fatty acid side chain, such as on the right, that double bond can cause a kink, which makes it easier for the body's enzymes to reach and digest those types of fats, and prevents those fatty acids from stacking up on each other. Though delicious. Butter and olive oil. Examples of saturated and unsaturated fatty acids. The cis bond being where the 2 bulky groups connected to the double bonded carbons are on the same side, and then we get that kink as shown. Fats made from saturated fatty acids are called saturated fats and are often solid at room temperature. Most animal fats are saturated. Fats made from unsaturated fatty acids are called unsaturated fats or oils and are often liquid at room temperature. Plant fat and fish fats are usually unsaturated, which tend to be healthy sources of unsaturated fats. A diet rich in saturated fats may contribute to cardiovascular disease through plaque deposits. Hydrogenation is the process of converting unsaturated fats to saturated fats by adding hydrogen across that double bond set of carbons. Hydrogenating vegetable oils also creates unsaturated fats with trans double bonds. These trans fats may contribute worse than saturated fats to the cardiovascular system. So typically cis trans fats or cis unsaturated fats are typically better than saturated fats, which are better than trans fats. In moderation. The major function of fats is to store energy. Humans and other mammals store their long-term food reserves as in adipose cells. Adipose tissue also cushions vital organs and also helps regulate temperature by insulating the body. A phospholipid is a type of fat where 2 fatty acids and a phosphate group are attached to a glycerol. Instead of 3 fatty acid chains, one of those is replaced with a phospholipid, or a phosphate group. 2 fatty acid tails are hydrophobic, but the phosphate group and whatever is attached is usually hydrophilic, which is useful for making cellular membranes, as in all cells. The phospholipid bilayer, the hydrophobic tails, like to face each other and exclude water, whereas the hydrophilic heads like to face the water either on the outside of a cell or the cytoplasm on the inside of a cell. When phospholipids are added to water, they self-assemble, sometimes into double-layered sheets called bilayers. At the surface of a cell, phospholipids are also arranged in a bilayer when the hydrophobic tails pointing toward the interior, and the phospholipid bilayer forms a boundary between the inside and outside of a cell, allowing for structure to remain inside the cell and separate from the external environment. Last class of fats include steroids, which are lipids characterized by a carbon skeleton consisting of 4 fused rings. Cholesterol is a type of steroid and is a component in animal cell membranes and a precursor from which other steroids are synthesized. A high level of cholesterol in the blood may contribute to cardiovascular disease. The basic structure in most of steroids is that 4 fused rings, 3 hexagons and 1 pentagon, and then whatever is attached to those can lead to a variety of functions. Getting into proteins, they account for more than 50% of the dry mass of cells. Some proteins speed up chemical reactions. Other protein functions include defense, storage, transport, cellular communication between different cells, the movement, and structural support. There are enzymatic proteins that help speed up reactions, such as the breaking up of polymers into separate monomers or separate units. There are defensive proteins, such as antibodies, which are able to find pathogens or help cells find pathogens. There are storage proteins. And then there are transport proteins. Transport proteins are embedded in cellular membranes, and they allow or aid in certain types of material crossing the boundary between the external environment and the internal environment of a cell. There are hormonal proteins. One of which being insulin, which helps reduce excess blood sugar. There are receptor proteins that exist on the outside of cells, or they help send chemical signals. There are contractile and motor proteins that are involved in movements, such as in muscles. And then there are structural proteins that have the function of support, which many examples can be found in connective tissues. Lots of different publisher videos. Enzymes and proteins that act as catalysts to speed up chemical reactions that might occur naturally in nature without enzymes, but could occur at a phenomenally slower rate if not for enzymes. The term catalyst means speeds up a chemical reaction. Enzymes can perform their functions repetitively without being destroyed. Therefore, they're reusable. Proteins are constructed from the same set of 20 naturally occurring amino acids. Polypeptides are unbranched polymers built from these amino acids. A protein is a biologically functional molecule that consists of one or more polypeptide chains. Amino acids are organic molecules with amino and carboxylic groups. Amino acids differ in the properties due to the 20 different naturally occurring amino acids differ in their chemical properties due to differing side chains called R groups. Each amino acid always starts out with an amino group, the nitrogen end, or the N terminus, and the carboxylic group, the carboxy end, or the C terminus. And then we have our alpha carbon in between. The R group is simply the only thing that differs between different types of amino acids. And the different types include those that have non-polar side chains that give a protein hydrophobic properties that pushes away from water. There are proteins that are hydrophilic or polar. They like to be near water. There are those that have charges, different polar chains, some that can be negatively charged and are usually acidic, and those that are positively charged side chains that can be basic. Therefore, owing to whether certain enzymes have portions of it that may like to avoid water and portions that like to be facing water, causing proteins to fold in unique ways, giving their structure, and some amino acids that give rise to whether an amino acid is only functional in certain pH or certain temperature ranges, from basic to acidic to low to middle to high temperatures. While there are 20, we'll go through the different types of naming. There's the full name, there's the 3-letter abbreviation, and then there's the single-letter abbreviation. Glycine, GLY or G, alanine, ALA or A, valine, which can be VAL or V, leucine, which can be LEU or L, isoleucine, which is ILE or I, methionine, which can be represented as MET or M, phenylalanine, PHE or F, tryptophan, TRP or W, proline, PRO or P. These are examples of the amino acids with non-polar side chains that are hydrophobic. Polar side chains, serine, SER or S, threonine, THR or T, cysteine, CIS or C, tyrosine, TIY, TYR or Y, asparagine, ASN or N, glutamine, GLN or Q. These are examples of hydrophilic carboxylic acid side chains that are polar. Then there are the electrically charged hydrophilic side chains. The negatively charged or acidic side chains are aspartic acid, ASP or D, and then glutamic acid, GLU or E, which only differ in the carbon backbone length. And then the positively charged or basic side groups include listine, LIS or K, arginine, ARG or R, and then histidine, HIS or H. Amino acids are linked by covalent bonds called peptide bonds. A peptide is a polymer of amino acids. Polypeptides range in length from easily a few to more than 1,000 monomers. Each polypeptide has a unique linear sequence of amino acids with a carboxylic end, the C terminus, or the amino end, the N terminus. When the polypeptide bond is formed, it's again through dehydration, condensation, dehydration synthesis, where a water molecule is lost, and then the C terminus of one chain, and then the N terminus of another chain or monomer are bound together chemically. And as you get different side chains grouped up together, that's how we will arise to different functional groups. The specific activities of proteins result from their intricate three-dimensional architecture. A functional protein consists of one or more polypeptides, precisely twisted, folded, and coiled into unique shapes. Now we're looking at the three-dimensional structures through three different types of models. There's the space-filling model, which shows the individual atoms as spheres. The ribbon model, which really just shows the chains and whether there are any alpha helixes or beta sheets. And then there's the wireframe model, which essentially shows all the individual amino acid chemical structures present. This is often seen in bioinformatics, should you take that course in a future program for your sophomore, or I should say junior or senior year in biology or biochemistry. The sequence of amino acids determines a protein's three-dimensional structure. A protein structure determines how it works. The function of a protein usually depends on its ability to recognize and bind to some other molecule, such as an antibody protein bonding to the protein from a flu virus. There are typically 4 levels of protein structure. Now those levels range from the primary structure, which is of a protein in its unique sequence of amino acids. The secondary structure, found in most proteins, that consists of the coils and folds in a polypeptide chain specifically. The tertiary structure, which is determined by the interactions of the side chains or R groups. And then there's quaternary structure, which results when protein consists of multiple polypeptide chains that wrap around together. So we're looking at the primary structure, which is just the chain of amino acids. Secondary structure, which is how does that particular chain fold, forming helixes or sheets, alpha helixes or beta sheets, where different hydrogen bonding can allow coils in the same alpha helix to be firm together, or different beta sheets across separate portions of a chain to be hydrogen bonded together. The tertiary structure, which is just how multiple alpha helixes and beta sheets come together. And the quaternary structure, which is how multiple polypeptide subunits can approach each other to form an entire protein. What we see here is that there are hydrophobic regions that essentially cause structure by pushing away from water. There could be disulfide bonds that form between 2 sulfur-containing amino acid side chains. There can be hydrogen bonding between different side chains across a polypeptide, and there can even be ionic bonding between charged amino acid side groups. What we're seeing here is the protein hemoglobin found in our red blood cells, which has iron heme centers which bind to oxygen. There are alpha and beta subunits, specifically 2 of each, which come together to form an entire hemoglobin protein. The primary structure of a protein is its sequence of amino acids. Primary structure is like the order of letters in a long word. Primary structure is determined by inherited genetic information, going all the way back to how our DNA nucleotides are sequenced and ordered. The coils and folds of secondary structure result from hydrogen bonding between repeating constituents of a polypeptide backbone. Typical secondary structure of a coil, called an alpha helix, and a folded structure called a beta pleated sheet. The tertiary structure, the overall shape of a polypeptide, results from interactions between R groups rather than interactions between backbone constituents. These interactions include hydrogen bonds, ionic bonds, hydrophobic interactions, and van der Waals interactions. Strong covalent bonds can form that are called disulfide bridges that help reinforce a protein's three-dimensional structure. Quaternary structure results when two or more polypeptide chains form one entire macromolecule. An example being collagen, a fibrous protein consisting of 3 polypeptides coiled like a rope. Then there's hemoglobin, is a globular protein consisting of 4 polypeptides, 2 of which called alpha and 2 called beta subunits. Sickle cell disease can result from a slight change in primary structure, where a slight change, even one amino acid difference, can often change the entire three-dimensional structure, which affects a protein's function, whether partly or entirely. Sickle cell disease and inherited blood disorder results from a single amino acid substitution in the protein hemoglobin. The abnormal hemoglobin molecules cause the red blood cells to aggregate into chains and deform into a sickle shape. What we hear is comparing the standard hemoglobin to sickle cell hemoglobin, resulting in the shape of the red blood cell to eventually change from more spherical, ovoidal, to sickle-shaped. In addition to primary structure, physical and chemical conditions can also affect structure. Alterations in pH, salt concentrations, temperature, or other environmental factors can cause a protein to unravel, which is called denaturing or denaturation, which is the loss of its inherent native three-dimensional structure. A denatured protein is usually biologically inactive because the three-dimensional structure is what gives it its function. After a protein is denatured, it is not always possible for it to renature or go back into its three-dimensional structure. It is hard to predict a protein structure from its primary structure. Most proteins probably go through several stages on their way to a stable structure. Diseases such as Alzheimer's, Parkinson's, and mad cow disease are associated with misfolded proteins that aren't in their healthy arrangement. Scientists can use X-ray crystallography to determine a protein's structure. Another method is nuclear magnetic resonance, called NMR spectroscopy, which does not require protein crystallization. NMR is the same principle as MRI scans, but in MRI, a magnet is spun around a patient. In NMR, a chemical sample is spun in a magnetic field, and usually the sample can be liquid or solid, whereas X-ray crystallography requires a very clean, solid, and specifically a crystal structure of a material. Bioinformatics is another approach to the prediction of protein structure and amino acid sequences by simply taking large pools of biological research and information and using computer technology to sort through it all to find meaningful connections. X-ray diffraction is a beautiful way to confirm the structure of something. The amino acid sequence of a polypeptide chain is programmed by a unit of inheritance called a gene. Genes are what tells the body how to make proteins, like in what order amino acids should be combined. Genes consist of DNA, a nucleic acid made of monomers called nucleotides. There are two types of nucleic acids. There's deoxyribonucleic acid and ribonucleic acid, shortened to DNA and RNA. DNA provides directions for its own replication. DNA directs synthesis of messenger RNA, and through messenger RNA controls protein synthesis. The process is called gene expression, taking information in a gene and eventually using it to make a protein. As a strand of messenger RNA is synthesized from a strand of DNA, in the case of eukaryotic cells, it will leave the nucleus and enter the cytoplasm, where a complex of a ribosome with ribosomal RNA can bind to the mRNA and then slowly build a polypeptide chain as amino acids are brought to the ribosome through transfer RNA. Each gene along a DNA molecule directs synthesis of a messenger RNA. The messenger RNA molecule interacts with the cell's proteins, synthesizing machinery to direct production of a polypeptide. The flow of genetic information can be summarized as DNA gets transcribed into RNA, which then gets translated into proteins. Nucleic acids are polymers called polynucleotides. Each polynucleotide is made of monomers called nucleotides. Each nucleotide consists of a nitrogenous base, a pentose sugar, and one or more phosphate groups. The portion of a nucleotide without the phosphate group is called a nucleoside. So once a nucleoside has a phosphate group, that is an entire nucleotide, which then go on to become DNA or RNA. So nucleoside, nitrogenous base plus the sugar. There are two families of nitrogenous bases, the pyrimidines, which include cytosine and thymine in DNA, or cytosine and uracil in RNA. They have a single 6-membered ring in their structure, but then there are purines, adenine and guanine, which show up in both DNA and RNA. They have a 6-membered ring fused to a 5-membered ring. In DNA, the sugar is deoxyribose, and in RNA, the sugar is ribose. A nucleotide is a nucleoside with the phosphate group. To reiterate, in DNA, different strands of DNA, cytosine likes to base pair with guanine on separate strands, and thymine likes to base pair with adenine on separate strands of DNA, although uracil in RNA likes to base pair from with adenine as RNA is being made. Main difference between deoxyribose and ribose is deoxyribose has a loss of the hydroxide group, hence deoxy, and whereas ribose still retains that hydroxide group on carbon 2. For the nitrogenous bases, both DNA and RNA has cytosine, adenine, and guanine, but thymine only shows up in DNA strands and uracil only shows up in RNA strands. Why? I'm not sure. Unless the body just wanted to be able to tell RNA and DNA apart. Interesting stuff. Alright, and here's just a structure of DNA. Base pairing, where we have two separate strands, and then RNA that was produced from one of those strands. RNA is typically single-stranded. Nucleotides are linked together by a phosphodiester linkage to build a polynucleotide. A polydiester linkage consists of a phosphate group that links the sugars in two nucleotides. The links create a backbone of sugar-phosphate units with nitrogenous bases and appendages. The linkage of bases along DNA or mRNA polymer is unique to each chain, each gene. DNA molecules have two polynucleotides spiraling around an imaginary axis, forming a double helix. The backbones run in opposite 5' to 3' directions from each other, an arrangement referred to as anti-parallel. One DNA molecule includes many genes. One DNA molecule can include multiple genes. Only certain bases in DNA will pair up and form hydrogen bonds. Adenine always base pairs with thymine, and guanine always base pairs with cytosine. This is called complementary base pairing. This feature of DNA structure makes it possible to generate two identical copies of each DNA molecule in a cell that is preparing to divide. Each DNA molecule being the separate strand of DNA. RNA, in contrast to DNA, is single-stranded. Complementary pairing can also occur between two RNA molecules and between parts of the same molecule. In RNA, thymine is replaced with the nucleotide uracil, so adenine and uracil pair up on separate strands. While DNA always exists as a double helix, RNA molecules are more variable in form. A unique form of ribonucleic acid mentioned before is tRNA or transfer RNA. And essentially, within the same strand, base pairing can occur at different portions of the strand, causing it to coil up. Once the structure of DNA and its relationship to amino acid sequence was understood, biologists sought to decode genes by learning their base sequences. The first chemical techniques for DNA sequencing were developed in 1970 and refined over the next 20 years. Amazing that some of this stuff is in our parents' lifetime. It is enlightening to sequence the full complement of DNA in an organism's genome. The rapid development of faster and less expensive methods of sequencing was a side effect of the Human Genome Project. Many genomes have been sequenced, generating large sets of data. Bioinformatics uses computer software and other computational tools to deal with the data resulting from sequencing numerous genomes within a species and across separate species, analyzing large sets of genes, or even comparing whole genomes of different species is called genomics. A similar analysis of large sets of proteins includes the sequences called proteomics, which are useful for making valuable connections, which can then be used in different studies. All right, we're almost wrapped up. Lots of figures. Sequences of genes and their protein products document the hereditary background of an organism. Linear sequences of DNA molecules are passed from parents to offspring. We can extend the concept of molecular genealogy to relationships between species. Molecular biology has added a new measure to the toolkit of evolutionary biology, essentially comparing similar strands of DNA sequences that can show up in even monkey and gibbons, or other species as well, to see similar alignments. And this DNA, this alignment that's being shown right now, is the single letter abbreviation for different amino acids. The same amino acid or the same protein may show up in multiple species, but there may be slight differences in how that protein is shaped owing to its various levels of function across different species. And then some final figures showing different structures of all the macromolecules we talked about. All right. As always, everyone, it is a pleasure to communicate with all of you. Welcome to the latest chapter, and, well, thank you for participating in this lecture in general biology. Peace.